Welcome to Biomolecules, the chemistry of life. In this presentation, we explore the four major classes of organic molecules that form the foundation of all living organisms: carbohydrates, lipids, proteins, and nucleic acids. Each of these biomolecules plays a critical and unique role in cellular structure, energy storage, catalysis, and genetic information transfer. Carbohydrates serve as energy sources and structural components. Lipids form membranes and store energy. Proteins carry out nearly every cellular function through their diverse structures. Nucleic acids store and transmit genetic information. Through detailed theory slides, interactive quizzes, and fully worked problems, you will gain a comprehensive understanding of these essential molecules that make life possible. All biomolecules are carbon-based organic compounds built from smaller monomer subunits. Carbohydrates are composed of monosaccharide units linked by glycosidic bonds. They function as immediate energy sources, energy storage, and structural components. Lipids are hydrophobic molecules that include fats, phospholipids, and steroids. They are primarily energy storage molecules and structural components of cell membranes. Proteins are polymers of amino acids linked by peptide bonds. They perform diverse functions, including enzymatic catalysis, transport, structural support, and cell signaling. Nucleic acids, DNA and RNA, are polymers of nucleotides that store and transmit genetic information. The central dogma of molecular biology describes the flow of genetic information from DNA to RNA to protein. Understanding these four classes provides the foundation for all of biochemistry and molecular biology. Carbohydrates are the most abundant biomolecules on Earth. They have the general formula CnH2ON, from which the name carbohydrate, meaning hydrated carbon, is derived. Carbohydrates are classified based on the number of sugar units. Monosaccharides are single sugar units like glucose, fructose, and galactose. They contain between 3 and 7 carbon atoms. Glucose has the molecular formula C6H12O6 and exists in both linear and cyclic forms. Disaccharides consist of 2 monosaccharide units joined by a glycosidic bond. Sucrose is glucose plus fructose, lactose is glucose plus galactose, and maltose is 2 glucose units. Polysaccharides are long chains of monosaccharide units. Starch is the energy storage polysaccharide in plants, consisting of amylose and amylopectin. Glycogen is the highly branched energy storage form in animals. Cellulose is a structural polysaccharide with beta-1, 4 glycosidic bonds that humans cannot digest. Chitin is a structural polysaccharide found in arthropod exoskeletons. Monosaccharides are classified by the number of carbon atoms. Trioses have 3 carbons, tetroses have 4, pentoses have 5, and hexoses have 6. The most common monosaccharides are hexoses, like glucose, fructose, and galactose. Glucose is an aldohexose containing an aldehyde functional group. Fructose is a ketohexose containing a ketone group. In aqueous solution, monosaccharides cyclize to form ring structures. Glucose forms a 6-membered pyranose ring, while fructose forms a 5-membered furanose ring. The glycosidic bond is formed between the anomeric carbon of one monosaccharide and a hydroxyl group of another through a condensation reaction. Alpha-1,4 glycosidic bonds link glucose units in starch, while beta-1,4 glycosidic bonds link glucose units in cellulose. The alpha versus beta configuration makes a profound difference. Starch is digestible by humans, while cellulose is not, because the enzymes required to break beta bonds are absent in the human digestive system. Lipids are a diverse group of hydrophobic or amphipathic molecules that are insoluble in water but soluble in nonpolar organic solvents. They are not polymers in the traditional sense. The major classes of lipids include triglycerides, phospholipids, and steroids. Triglycerides consist of one glycerol molecule bonded to 3 fatty acid chains through ester linkages. Fatty acids are long hydrocarbon chains with a terminal carboxyl group. Saturated fatty acids have no double bonds and are solid at room temperature like butter. Unsaturated fatty acids contain one or more double bonds, introducing kinks in the chain that prevent tight packing, making them liquid at room temperature like olive oil. Phospholipids are the primary components of cell membranes. They have a polar hydrophilic head group containing phosphate and two hydrophobic fatty acid tails. In aqueous environments, phospholipids spontaneously form bilayers. Steroids have a characteristic four-ring fused carbon structure. Cholesterol is a vital steroid that modulates membrane fluidity and serves as a precursor to steroid hormones like estrogen and testosterone. The fluid mosaic model describes the cell membrane as a dynamic structure where phospholipids and proteins can move laterally within the bilayer. The amphipathic nature of phospholipids drives spontaneous self-assembly into bilayers in aqueous environments. The hydrophilic heads face the aqueous extracellular and intracellular environments, while the hydrophobic tails face inward, creating a hydrophobic barrier that controls molecular passage. Cholesterol is embedded within the membrane and modulates fluidity. At high temperatures, it restricts movement, and at low temperatures, it prevents tight packing. Saturated fatty acids pack tightly, making membranes less fluid, while unsaturated fatty acids with cis double bonds introduce kinks that increase fluidity. Lipids also serve as signaling molecules. Eicosanoids derived from arachidonic acid function as local hormones. Steroid hormones like testosterone and estrogen regulate gene expression. Lipids are also crucial for thermal insulation, protection of vital organs, and as carriers for fat-soluble vitamins A, D, E, and K. Proteins are linear polymers of amino acids. There are 20 standard amino acids, each consisting of a central alpha carbon bonded to an amino group, a carboxyl group, a hydrogen atom, and a variable R group side chain. The R group determines the chemical properties of each amino acid. Amino acids are classified as nonpolar hydrophobic, polar uncharged, acidic negatively charged, and basic positively charged. Hydrophobic amino acids like valine and leucine tend to cluster in the protein interior. Polar and charged amino acids are usually found on the protein surface. Peptide bonds form between the carboxyl group of one amino acid and the amino group of another through a condensation reaction, releasing water. The peptide bond has partial double bond character due to resonance, which restricts rotation around the CN bond and makes the peptide bond planar. The sequence of amino acids in a protein is called its primary structure. The polypeptide chain has directionality. The N-terminus has a free amino group, and the C-terminus has a free carboxyl group. Protein structure is organized into four hierarchical levels. Primary structure is the linear sequence of amino acids linked by peptide bonds, determined by the genetic code. Secondary structure refers to local folded structures stabilized by hydrogen bonds between backbone carbonyl and amide groups. The two most common secondary structures are the alpha helix, where the polypeptide chain coils into a right-handed spiral stabilized by hydrogen bonds between every fourth amino acid, and the beta-pleated sheet, where adjacent polypeptide segments align side by side, forming hydrogen bonds between them. Tertiary structure is the overall three-dimensional folding of a single polypeptide chain, stabilized by hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. The tertiary structure creates the functional active site of enzymes. Quaternary structure involves the assembly of multiple polypeptide subunits into a functional protein complex. Hemoglobin is a classic example. It consists of 4 subunits, 2 alpha and 2 beta, that work cooperatively to bind and release oxygen. Protein misfolding can lead to diseases such as Alzheimer's and Parkinson's. Nucleic acids are polymers of nucleotides that store and transmit genetic information. Each nucleotide consists of 3 components: a nitrogenous base, a 5-carbon pentose sugar, and a phosphate group. The nitrogenous bases are classified as purines, which have a double ring structure and include adenine and guanine, and pyrimidines, which have a single ring structure and include cytosine, thymine, and uracil. DNA contains deoxyribose sugar and uses thymine, while RNA contains ribose sugar and uses uracil instead of thymine. Nucleotides are linked by phosphodiester bonds between the 3' hydroxyl group of one sugar and the 5' phosphate group of the next. DNA is a double helix where 2 antiparallel strands are held together by hydrogen bonds between complementary base pairs. adenine pairs with thymine through 2 hydrogen bonds, and guanine pairs with cytosine through 3 hydrogen bonds. The sugar phosphate backbone runs along the outside of the helix, while the bases stack in the interior. RNA is typically single-stranded and has a more diverse set of functions, including messenger RNA, transfer RNA, and ribosomal RNA. The Watson-Crick model of DNA describes a right-handed double helix with a diameter of approximately 2 nanometers. The two strands run antiparallel, meaning one runs in the 5' to 3' direction, while the other runs 3' to 5'. The sugar phosphate backbone is hydrophilic and faces outward, while the nitrogenous bases are hydrophobic and stack in the interior. The base pairs are nearly planar and perpendicular to the helix axis. There are approximately 10 base pairs per complete turn of the helix. The major groove and minor groove provide binding sites for proteins that regulate gene expression. DNA replication is semi-conservative. Each parental strand serves as a template for a new complementary strand, catalyzed by DNA polymerase. The genetic code uses 3 nucleotide codons to specify each amino acid. There are 64 possible codons for 20 amino acids, making the code degenerate. The central dogma describes the flow of information. DNA is transcribed into messenger RNA, which is translated into protein by ribosomes. This fundamental process underlies all of life. Quiz question 1. Which of the following carbohydrates is a polysaccharide used for energy storage in animals? Option A, cellulose. Option B, starch. Option C, glycogen. Option D, chitin. Take a moment to think about your answer. The correct answer will be revealed shortly. Quiz question 2. Which of the following best describes phospholipids in aqueous solution? Option A, they form micelles with tails outward. Option B, they spontaneously form bilayers. Option C, they dissolve completely in water. Option D, they precipitate as solid crystals. Consider the amphipathic nature of phospholipids. The answer will appear in a moment. Quiz question 3. Which level of protein structure is determined solely by the sequence of amino acids? Option A, primary structure. Option B, secondary structure. Option C, tertiary structure. Option D, quaternary structure. Think about which level is directly encoded by DNA. The answer is coming up. Quiz question 4. According to Chargaff's rules, if a DNA sample contains 30% adenine, what is the guanine content? Option A, 30%. Option B, 20%. Option C, 40%. Option D, 70%. Remember the base pairing rules? The correct answer will be shown shortly. Quiz question 5. Which type of bond links amino acids together in a protein chain? Option A, glycosidic bond. Option B, phosphodiester bond. Option C, peptide bond. Option D, hydrogen bond. This is a fundamental concept in protein chemistry. The answer will be revealed soon. Problem 1. Carbohydrate formula. A monosaccharide contains 40% carbon, 6.71% hydrogen, and 53.3% oxygen by mass. Its molar mass is 180.15 grams per mole. Find its molecular formula. Step 1. Calculate the mass of carbon. 0.400 times 180.15 equals 72.06 grams. Step 2. Hydrogen mass. 0.0671 times 180.15 equals 12.09 grams. Oxygen mass. 0.533 times 180.15 equals 96.02 grams. Step 3. Moles of carbon. 72.06 divided by 12.01 equals 6.00. Moles of hydrogen. 12.09 divided by 1.008 equals 12.00. Moles of oxygen. 96.02 divided by 16.00 equals 6.00. Step 4. The mole ratio C to H to O is 6 to 12 to 6, giving the empirical formula CH2O. Step 5. N equals 180.15 divided by the empirical formula mass of 30.03, which equals 6.00. Step 6. The molecular formula is CH2O times 6, giving C6H12O6. Problem 2. Fatty acid saturation. Palmitic acid is a saturated C16 fatty acid with molar mass 256 grams per mole. Oleic acid is monounsaturated C18:1 with molar mass 282 grams per mole. Compare iodine numbers and explain melting point differences. Step 1. Palmitic acid has 0 double bonds, so its iodine number is 0. Step 2. Oleic acid has 1 cis double bond at carbon 9. Step 3. Iodine moles per mole of oleic acid equals 1. Mass of I2 is 253.8 grams. Step 4. Iodine number is 253.8 divided by 282 times 100, which equals 90.0. Step 5. Palmitic acid melts at 63 degrees Celsius because saturated chains pack tightly. Step 6. Oleic acid melts at -2 degrees Celsius because the cis double bond kink prevents tight packing. Problem 3. Peptide formation. Glycine and alanine form a dipeptide. Calculate the molar mass of the dipeptide and the water released. Step 1. Glycine C2H5, NO2 has molar mass 75.07 grams per mole. Step 2. Alanine C3H7, NO2 has molar mass 89.09 grams per mole. Step 3. Glycine plus alanine forms gliolidipeptide plus water. Step 4. Dipeptide mass equals 75.07 plus 89.09 minus 18.02, which is 146.14 grams per mole. Step 5. Exactly one water molecule of 18.02 grams per mole is released. Step 6. For a tripeptide, 2 water molecules are released. An N-peptide releases N-1 water molecules. Problem 4. DNA base pairing. A double-stranded DNA fragment has 2,400 base pairs. If 30% of the bases are cytosine, how many adenine bases are present? Step 1. Cytosine is 30%, so guanine is also 30% by Chargaff's rule. Step 2. G plus C equals 60%, so A plus T equals 40%. Step 3. Since A equals T, adenine is 20%. Step 4. Total bases equals 2,400 base pairs times 2, which is 4,800 individual bases. Step 5. Number of adenine bases is 0.20 times 4,800, which equals 960. Step 6. Check. G equals 1.440, C equals 1.440, A equals 960, T equals 960. T equals 960. Total. 1440 plus 1440 plus 960 plus 960 equals 4,800.